everyone, welcome back to another video of DNA Bioscience. Today we are going to learn about the primary and secondary immune response. So immune response, as you all know, that immune response is designed in such a way that they can act as a defense mechanism. They can fight and prevent ourselves from the infection. So let's discuss the primary immune response. The primary immune response occurs when an antigen comes in contact to the immune system for the first time. So when an antigen comes in contact to the immune system first time, then the primary immune response occurs. During this time, the immune system has to learn to recognize the antigen, how to make antibody against it, and eventually produce memory lymphocytes. So, this is the primary response diagram. It consists of a lag phase. The lag phase is when an antigen is exposed first time to the body, then it takes some time to develop the antibodies against it. So this is the lag phase where no antibodies are present. Then the next phase is the exponential phase or the log phase, where higher number of antibodies are produced. Then the next phase is plateau phase or the steady phase, where the antibody level or the concentration remains constant. And the next phase is the decline phase, where the antibody concentration declines. So in the x-axis, antibody concentration in the serum is mentioned, and in the y-axis the days it takes to produce the response. So after 5 days the antibody production generally occur. In the lag phase there is no antibody production. So we can say after 1 week delay the antibody production occurs, and the antibodies, they have a low affinity in case of primary response, and they are of very short life for only weeks. You can see in this picture, this is the 15 days and the antibody is constant in this, and this is the decline phase, so it lasts for only few weeks. And the predominant antibody present in the primary response is IgM type. The next is the secondary immune response. Secondary response occurs when the same antigen enters the body again, maybe the second time or third time. So here the lag phase, you can see it's a very small phase, and the exponential phase is very rapid and higher level of antibody production takes place. And in this case the predominant antibody is IgG type. So in the primary response, when an antigen is exposed first time to the body, that is called the primary dose. But in the secondary response, high levels of antibody produced by subsequent injection of the antibody, that is called the booster dose. So let's summarize it once again. The primary response, it is divisible into 4 phases: lag phase, exponential phase, steady phase, and decline phase. The lag phase, the initial exposure of antigens are detected in the blood and takes about a week. In this time, the specialized B and T cells are activated by contact with the antigen. In the exponential phase, there is a sharp rise in the levels of antibodies which are secreted by large numbers of plasma cells. In the steady state, the antibody levels stay relatively constant due to continuous secretion of antibodies to replenish any that have degraded. And in the declining phase, antibody levels slowly decrease due. So let's discuss the differences. The first exposure to a specific antigen, and it is the second exposure to the same antigen. It takes 1 week. It occurs within hours. The affinity of antibodies are very low, in this case very high. The response is generally smaller, the response is larger. They are activated by the immune genes, and the secondary immune response is activated by the peptide antigen. The process is very short and it takes only few weeks. Secondary response forms antibodies for many months. And the predominant antibody in the primary response is IgM, and in the secondary response is IgG. So that's all about the primary and secondary immune response. I hope it will help you. Thank you.